Hormones, general mechanism of hormonal action. Higher animals and human beings possesses a diversity of cells, tissues, organs, and systems for specialized functions. They require mechanisms by which different cells can communicate with each other. Intercellular communication occurs through nerve system and endocrine system. Nerve system and endocrine system also communicate with each other. Nerve system, electrical chemical signals transmitted through mixed structural roots, and endocrine system, chemical signals transmitted in the form of mobile hormone molecules. External or internal signal, hypothalamus with the help of releasing hormone, anterior pituitary, then trophic hormone, then target endocrine gland, then ultimate hormone. Along with that, systemic effects will be produced. Once again, external or internal signal, hypothalamus, anterior pituitary, target endocrine gland, ultimate hormone, and systemic effects. So hormones are present in blood in very small concentrations, yet they produce profound biological effects. The action of a hormone occurs through a cascade of events in which the signal is amplified at a number of stages. Here you could see an example: glucagon receptor complex. Glucagon receptor complex, inactive adenylate cyclase, active adenylate cyclase, then amplification will be taking place. ATP changes to CAMP, then inactive protein kinase A, then active protein kinase A, there will be amplification again. So inactive phosphorylase kinase B turn into active phosphorylase kinase B, then that will lead to amplification again, then inactive phosphorylase turn to active phosphorylase. Later there will be amplification. Along with that, glycogen transfer into glucose 1P, then glucose. A molecule secreted by some cells that controls and regulates the activity of certain cells or organs. Hormone definition: secreted by some cells that controls and regulates the activity of certain cells or organs. The hormone acts on its target cells, which possess specific receptors for the hormone. Then the target cells may be distant or nearby. Then, depending on the distance between the target cell and the hormone secreting cell, the hormone may be endocrine hormone, paracrine hormone, autocrine hormone. The endocrine hormones are secreted by endocrine ductless glands and act on distant cells. The paracrine hormones act on cells in near vicinity of the hormone secreting cells. The autocrine hormones act on the cells secreting the hormone. Then hormone receptors. Target cells possesses receptors for hormones. Chemically, the receptors are proteins. A receptor is specific for a particular hormone. It has at least two domains, a recognition domain and a signal domain. The receptor may be located in the cell membrane or inside the cell. Then the intracellular receptors may be located in the cytosol or in the nucleus. A given cell may possess receptors for several hormones. Here you could see a pictorial depiction. Nucleus is there, cell membrane. So here you could see 1, 2, 3, 4, 5, 6 are hormone receptors. See where are they located. Okay. The number of receptors is not fixed. The receptors can undergo up regulation, increase in the number of receptors, down regulation, decrease in the number of receptors. Classification of hormones. Hormones may be classified on the basis of chemical nature, solubility, location of receptors, and nature of second messengers. On chemical basis, hormones can be divided into proteins, peptides, and amino acid derivatives, and steroids. Then on the basis of solubility, hormones can be divided into hydrophilic hormones. That means water loving, like proteins, peptides, and catecholamines. Then hydrophobic hormones. Hydrophobic in the sense what are hating. Steroid hormones actually and thyroid hormones. Remember hydrophilic hormones, example protein, peptide and catecholamines. Hydrophobic hormones, steroid hormones and thyroid hormones. And the classification based on the location of receptors and nature of second messengers is more comprehensive. On this basis, the hormones may be divided into two groups. Group 1 hormones and group 2 hormones. Group 1 hormones, these hormones are lipophilic. Examples are steroid hormones, thyroid hormones, calcitrol, etc. Being lipophilic, they require carrier proteins to transport them in blood. Then the receptors for glucocorticoids and mineralocorticoids are present in the cytosol. Hydrophilic hormones, proteins, peptides and catecholamines. Hydrophobic hormones, steroid hormones and thyroid hormones. Then the classification based on the location of receptors and nature of second messengers is more comprehensive. On this basis, the hormones may be divided into two groups on the basis of location of receptors and nature of second messengers. Group 1 hormones and group 2 hormones. First, group 1 hormones. Group 1 hormones are lipophilic. Examples are steroid hormones, thyroid hormones and calcitrol. Lipophilic in the sense lipid loving. That means most probably they are going to be hydrophobic. Okay, so these hormones are lipophilic. Examples are steroid hormones, thyroid hormones, calcitrol, etc. Being lipophilic, they require carrier proteins to transport them in blood. The receptor for glucocorticoids and mineralocorticoids are present in the cytosol. The receptors for estrogen, progesterone, thyroid hormones and calcitrol are present in the nucleus. Then mechanism of action of group 1 hormones. Receptors of group 1 hormones are intracellular. Receptors of group 1 hormones are intracellular. Being lipid soluble, the hormones can easily traverse the cell membrane. Being lipid soluble, the hormones can easily traverse the cell membrane. Then the hormones bind to their intracellular receptors. The hormone receptor complex is mobile. It does not require any second messenger to carry the signal. Wait a minute. The hormone bind to their intracellular receptor. The hormone receptor complex is mobile. It does not require any second messenger to carry the signal. Then the receptor possesses one domain to recognize and bind to the hormone, another domain to recognize a specific sequence in the DNA. The DNA sequence is called hormone response element. Hormone response element, HRE, hormone response element. There are different response elements for different hormones like glucocorticoid response element, mineralocorticoid response element, thyroid hormone response element. The HREs are located just upstream of some structural genes and their promoters. The hormone receptor complex goes to the nucleus and binds to the HRE. This increases the transcription and translation of specific genes. The proteins synthesized produce the biological effects attributed to the hormone. For example, glucocorticoids increase the expression of genes encoding gluconeogenic enzymes. Increased synthesis of these enzymes increase gluconeogenesis. Hormone receptor complex, HRE, the nucleus, DNA, mRNA, ribosome, protein, biological effect is produced. Group 2 hormones include proteins, peptides and catecholamines. Being hydrophilic, they do not require carriers to transport them in blood. Their receptors are transmembrane proteins. Their receptors are transmembrane proteins. The hormone binds to its receptor on the cell surface and does not enter the cell. The second messenger is required to carry the message inside the cell. The second messenger is produced by an effector. Then mechanism of action of group 2 hormones. On hormone receptor binding, a signal transducer is activated. The signal transducer goes to and activates an effector. The effector produces a second messenger. Second messenger includes cyclic AMP, CAMP, cyclic GMP, cyclic GMP or CGMP, CA2 plus or phosphorinositides, then tyrosine kinase. These are examples of second messengers. Then CAMP. The hormones using CAMP as a second messenger include TSH, ACTH, FSH, glucagon, epinephrine acting through the alpha 2, beta 1 and beta 2 adrenergic receptors. CAMP is produced in the cell from ATP. CAMP is produced in the cell from ATP by adenylate cyclase, a membrane-bound enzyme that is normally inactive. CAMP is produced in the cell from ATP by adenylate cyclase, a membrane-bound enzyme that is normally inactive. ATP, adenylate cyclase, CAMP plus PPA. Binding of the hormone to its receptor activates adenylate cyclase. Concentration of CAMP in the cell increase. This produces the biological effect. Most of these effects occur due to CAMP-induced activation of protein kinase A. For example, activation of protein kinase A in liver by glucagon increase glycogenolysis, decrease glycogenesis. Then adenylate cyclase effector is not in contact with the hormone receptor. The signal is carried from the receptor to the effector by the signal transducer, receptor effector coupling. The signal transducer is a G protein. Receptor G protein. G protein is a trimer made up of an alpha subunit, a beta subunit and gamma subunit. The alpha subunit has a site which can be occupied by GDP or GTP. Normally it is occupied by GDP. And then hormone binds to its receptor, GDP is displaced by GTP. Then the GTP bearing alpha subunit dissociates from the beta and gamma subunits. The alpha subunit goes and binds to adenylate cyclase. Adenylate cyclase is activated. Active adenylate cyclase produces CAMP. Then cell membrane receptor G protein hormone adenylate cyclase GTP form change. Then alpha subunit has intrinsic GTPase activity which slowly hydrolyzes GTP to GDP. When GTP is converted into GDP, the alpha subunit dissociates from the effector. It goes back to and reassociates with the beta and gamma subunits. Then the G proteins which stimulate the effector are known as stimulatory G proteins or GS proteins. There are inhibitory G proteins or GI proteins also which inhibit the effector. Then CGMP. The hormones using CGMP as a second messenger include atrial natriuretic peptide ANP, nitric oxide, and CGMP is formed from GTP by guanylate cyclase. Then GTP, PPI guanylate cyclase, CGMP phosphodiesterase GMP. Two forms of guanylate cyclase are known: membrane bound form, cytosolic soluble form. ANP activates the membrane bound form. NO activates the cytosolic form. Guanylate cyclase activity is present in the cytoplasmic portion of the ANP receptor. This switches on when AMP binds to its receptor on the cell surface. CAMP is secondary messenger for NO also. NO is the only hormone known so far which is a gas. NO is synthesized from arginine by the nitric oxide synthetase NOS. Guanylate cyclase activated by ANP and NO converts GTP into CGAMP. So CGMP activates protein kinase G. Protein kinase G phosphorylates some target proteins. This produces the biological effects. Atrial natriuretic peptide, cell membrane, guanylate cyclase domain, GTP, GMP, AMP receptor, protein kinase, phosphorylation of Soluble guanylate cyclase, phosphorylation of target protein, biological effects. Nitric oxide synthetase has three isoforms: INOS, NNOS, and ENOS. INOS is inaudible, inducible, and is present in macrophages and neutrophils, and NNOS is present in neutrons. ENOS is present in endothelial cells. NO synthesized in phagocytes aids in the destruction of phagocyte-sourced cells. NO synthesized in neurons acts as a neurotransmitter. NO formed in endothelial cells diffuse into adjacent smooth muscles. It causes vasodilation by relaxing the smooth muscles. Then hormones using Ca++ or phosphoinositides are second messenger include oxytocin, gastrin, cholecystokinin, then epinephrine acting through alpha-1 adrenergic receptors. Then binding of these hormones to their receptors activate phosphoinositidase. Phosphoinositidase is a membrane-bound enzyme. It is also known as phospholipase C. Signal is transmitted from the receptor to phospholipase C by a G protein. Activated phospholipase C acts on phosphatidylinositol 4,5-biphosphate. PAP2 is present in the cell membrane. It is formed by phosphorylation of phosphatidylinositol. Phospholipase C hydrolyzes phosphatidylinositol 4,5-biphosphate PAP3 into inositol triphosphate IP3 and diacylglycerol DAG. IP3 releases calcium ions from its intracellular stores. Calcium ions combine with calmodulin and activate calmodulin kinase. DAG activates protein kinase C. So activated calmodulin kinase and protein kinase C cause phosphorylation of some target proteins. The phosphorylated proteins produce the biological effects. Tyrosine kinase. This hormone acting via tyrosine kinase include insulin, growth hormone, prolactin, etc. These hormones act by phosphorylating tyrosine residues of some target proteins in the cell. Most of the growth factors also act by tyrosine phosphorylation. For example, insulin-like growth factor 1 IGF-1, insulin-like growth factor 2 IGF-2, nerve growth factor NGF, epidermal growth factor EGF, platelet-derived growth factor PDGF. So receptors for hormones acting via tyrosine kinase are transmembrane. The extracellular portion of the receptor has got the hormone binding domain. The cytoplasmic portion has got tyrosine kinase domain. When the hormone binds to the receptor, tyrosine kinase domain becomes active. Tyrosine kinase phosphorylates some tyrosine residues in the receptor itself. The receptor becomes autophosphorylated. The autophosphorylated receptor phosphorylates the tyrosine residues of some target proteins. The target proteins become active and produce the biological effects. An example of this subgroup of hormones is insulin. Insulin receptor is made of two alpha chains and two beta chains. The alpha chains are linked to each other by disulfide bonds. The alpha chains are linked to beta chains by disulfide bonds. The alpha chains are entirely extracellular. The beta chains are partially extracellular, partly membrane bound embedded, and partly cytosolic. The alpha chains possess the insulin binding site. The cytoplasmic portion of beta chains possess tyrosine kinase domain. The tyrosine kinase domain is normally active. Binding of the insulin to the alpha chain switches on the tyrosine kinase activity of beta chains. Active tyrosine kinase phosphorylates some tyrosine residues in the beta chains. The receptor becomes autophosphorylated. The autophosphorylated receptor phosphorylates tyrosine residues of some target proteins. This include insulin receptor substrate 1 IRS1, insulin receptor substrate 2 IRS2, and this initiates a cascade of reactions culminating in the varied biological effects of insulin. Then regulation of hormone secretion. Minute amount of hormones produce profound biological effects. Therefore, regulation of hormone secretion should be very precise. Hormone secretion should increase when its biological effect is required. It should decrease when the desired biological effect has been activated. Then hypothalamo-hypophyseal endocrine axis is one regulatory cascade. It is involved in the regulation of several hormones. Feedback inhibition is another important mechanism. A rise in hormone concentration causes feedback inhibition of hormone secretion. As an example, secretion of thyroid hormones is regulated by both the mechanisms. When there is need 4 thyroid hormones. Hypothalamus secretes thyrotropin releasing hormone. TRH stimulates anterior pituitary to secrete thyroid stimulating hormone. TSH acts on thyroid gland and stimulates the secretion of thyroid hormones. Raised blood level of thyroid hormones cause feedback inhibition of TRH secretion, TSH secretion, and secretion of thyroid hormones.